So before we start to talk about bacteria and the structure of the bacterial cell wall, it is important to remember that bacteria are not the only type of microorganism. So any organisms that we can see, if they're unicellular, for example, with an electron microscope, we're going to call them microorganisms. Now, if you pause the video, you can maybe have a quick go at this activity to try and identify the different types of microbes. Now, 3 of these will be living cells that can replicate, and one of them is a non-living pathogenic particle. So if you had a quick go at that activity, you should hopefully have seen that cell number A is a yeast cell. Now, we know yeast cells are eukaryotic. That means they have mitochondria inside them, so they can respire aerobically when there's oxygen present, so the mitochondria make ATP. We also know, obviously, yeast cells can undergo alcoholic fermentation when there's a lack of oxygen present. So that would not involve the mitochondria, that would just involve the enzymes in the cytoplasm, and ethanol and carbon dioxide would be produced when there's a lack of oxygen. Now, the outside of the yeast cell we know has a chitin cell wall. So that's quite different to plant cells, which have cellulose cell walls, and we know bacteria have peptidoglycan cell walls. Now, on the right hand side of this yeast cell, we can see a little bud having formed. Now, inside this bud is cytoplasm and replicated organelles like mitochondria, and eventually a nucleus with the same genetic code as the parent yeast cell on the left. Now, as the bud gets bigger, when it reaches the same size as the parent yeast cell on the left, the connection is severed, and that releases the new yeast cell into the environment. And what that does, it leaves on the parent cell what's called a budding scar. So as you can probably imagine, yeast cells can only bud a certain number of times because on their surface it leaves these little scars. Now, B was a bacterium, so we can see in the background this structure here. This bacterial cell is undergoing binary fission. So we know bacteria, they can, in this particular case, they can elongate. So they have to make a brand new cell wall made of peptidoglycan, and they have to replicate their DNA. And as they elongate, they then divide down the middle, and that produces 2 identical bacterial cells. So this is a bacillus bacterium. They're normally about 1 to 10 micrometers in length, depending on the species. Normally 1 to 5 is more typical. Now, the yeast cells on the left, they can be between 3 to 4 micrometers in diameter quite often. Depends on the type of yeast, on the species. They can be up to 30 micrometers in diameter, but that's more typical. So we know bacteria, they may have flagella. So we can see here at the back, there are multiple flagella, and they allow this particular bacterium to be motile, so it can swim in the extracellular fluid. That's the fluid on the outside of the cell. But inside these bacteria, there are no organelles, membrane-bound organelles. There's no nucleus, and there's no mitochondria. There's no rough ER or smooth ER. It's just cytoplasm with plasmids and DNA, circular DNA, that is free in the cytoplasm. Now, C at the bottom is a paramecium. Now, that belongs to the Protoctista kingdom. In particular, it's a type of protist. That's a unicellular Protoctista. Now, paramecium is eukaryotic, so it has organelles, membrane-bound organelles within inside it in its cytoplasm. You can just about see here in purple, that's what we call a food vacuole. So these paramecium can take in food particles from the medium that surrounds the cells. So these quite often are what we're going to call heterotrophic organisms. Just like yeast cells are, and many bacteria are heterotrophic because they use ready-made organic molecules and they ingest them, they take them in, and then they break them down, and then they utilize them for various processes. So paramecium are eukaryotic, they've got membrane-bound organelles within them, and they are unicellular. Now, D on the right-hand side at the bottom is a virus. Now, a virus is a non-living particle. They are pathogenic, and they can cause infection only if they bind to a receptor molecule on the surface of a host cell inside which they can then replicate. So a virus has quite a simple structure. It has no cytoplasm, it has no organelles, it has an outside protein coat called the capsid. So you can see in this picture, the capsid is on the outside. You've got these protein spikes here. So the spikes are made of different materials called hemagglutinin. You don't need to know that just yet for this topic. And this is the protein capsid coat. Now inside the virus is one of two types of nucleic acid. It's either DNA or it's RNA. Now most viruses that infect animal cells and plant cells are RNA viruses. They're called retroviruses. So things like influenza, HIV and herpes are retroviruses. And inside there might be one or more RNA molecules. So what happens is if the virus gets anywhere near a host cell, quite often these molecules on the capsid, they can actually bind to receptors on the surface or the cell membrane of the host cell. So they're complementary in shape, and this allows the virus to be taken into the host cell. The protein capsid coat dismantles and that releases the RNA. The RNA is then replicated to make many copies of that RNA inside the cytoplasm of the host cell, and the RNA can attach to ribosomes, and it uses the host machinery to translate the RNA at the ribosome of the host cell into viral proteins, i.e. new capsid proteins. So many new virus particles can form inside the host cell using its own protein making machinery, the ribosomes. So before we start to talk about the structure of the bacterial cell wall, it's important to talk about the positives of bacterial use and also some of the negatives. So we know bacteria are used in the food production industry, for example, to make cheeses. Now a cheese maker will always start obviously with milk, and to that milk they add a collection of enzymes called rennet. Now young mammals we know feed off milk, and the rennet enzymes are produced in the stomach of the mammal. Now the enzymes contain proteases, so the milk starts to get broken down, the proteins within the milk get broken down, and that causes the milk to thicken. So in these young mammals, as the milk passes down the small intestine or the rest of the gut, it becomes coagulated and thickened. Now cheesemakers use the enzymes. They don't these days get the enzymes from young mammals, they actually get them from genetically engineered bacteria. So they add the rennet enzymes to the milk. They also add another type of bacterium. So lactobacillus is a common type. And as the name suggests, these lactobacilli bacteria, they absorb lactose disaccharides from the milk. They also make lactase. So inside the bacterial cell, the lactase enzyme breaks down the lactose to glucose and galactose. And these monosaccharides are then used in respiration inside these lactobacillus bacteria. Now these are facultative anaerobe bacteria, and that means they will replicate in the presence of oxygen. They respire aerobically to make ATP, but they can also replicate in the absence of oxygen, and they'll switch to anaerobic respiration. So one product of anaerobic respiration in these lactobacillus bacteria is lactic acid. So lactic acid, and that's from lactic acid fermentation when there's no oxygen present. Now another use of bacteria is in genetic engineering. So inside the cytoplasm, we'll see next, in a bacterium, they have small circular pieces of DNA called plasmids. Now these plasmids can be extracted and human genes can be inserted into the plasmid to create a recombinant plasmid. Now these recombinant plasmids can be placed back into bacteria, and when these bacteria replicate by binary fission, they also replicate the DNA of the plasmid. So these plasmids with the human insert, the human gene inserted into them, they will get their proteins expressed. So these bacteria secrete lots and lots of human insulin. Now this insulin can be isolated and purified and turned into a medication. So insulin is a 10 amino acid polypeptide. So it's a very short polypeptide of only 10 amino acids. But it's important as a medication because in diabetics, in particular type 1 diabetics, they don't make their own insulin. Now, insulin is a very important hormone inside our bodies because it stimulates every cell of the body to absorb glucose from the bloodstream. So if you can't make your own insulin, you have a very high blood glucose level concentration in the blood, which can cause all sorts of damage to the tissues and cells of the body. Now, the way insulin works is that when it binds to its specific receptor on the surface of every cell in your body, it upregulates the glucose carrier protein in the membranes of those cells. So when insulin binds to its receptor, it causes certain messages to be sent to the nucleus. The same cell upregulates and makes more of the glucose carrier proteins, so these cells can then start to absorb glucose from the bloodstream. Now, we know from last year another function of bacteria is a mutualistic relationship between cellulase producing bacteria in the rumen, which is chamber number 1 of ruminant animals like the cow. So the cow has a 4-chambered stomach and the rumen is chamber number 1. So we're going to say here chamber 1. Now, the diet of the cow is obviously largely grass, so it's largely cellulose based, but the cow does not make cellulase enzymes. So these bacteria, it's a symbiotic or mutualistic relationship because they secrete cellulase enzymes, and this breaks down or hydrolyzes the glycosidic bonds in the cellulose, and it releases beta glucose, which we know is a monosaccharide. Now, these bacteria, they actually utilize some of the beta glucose and they convert it into what we call organic acids. So these organic acids are then absorbed through the rumen wall into the blood of the cow, and it can use the organic acids in respiration to release energy inside its cells. Now, another type of bacterium is Nitrosomonas and Nitrobacter, and these are involved in the nitrogen cycle. Now, we know any dead organism, whether it be a plant, an animal, or any other type of organism, they contain organic nitrogen within them. So the organic nitrogen is molecules like DNA or RNA, the nucleic acids. It could be the nucleotides, which are the subunits of the nucleic acids, or it can be things like amino acids, proteins, ATP, etc., chlorophyll. So saprophytes convert the organic nitrogen into ammonium ions. Now, this part of decomposition is called ammonification. So when we get dead organic matter being decomposed by saprophytes, we get an increase in ammonium ions in the soil. Now, Nitrosomonas absorbs the ammonium ions and converts these into nitrite, NO2- ions. Now, some of these nitrite ions are secreted into the soil and converted, or absorbed and then converted into NO3- nitrate ions by Nitrobacter. Now, these are both obligate aerobes. That means they only replicate in the presence of oxygen, so they respire aerobically. But we know when there's an increased nitrate content in the soil, then the soil becomes very fertile. We know all plants can absorb nitrate ions by active transport through their roots, and so it allows the plant to then synthesize its own organic nitrogen molecules, like we mentioned, DNA, RNA, the nucleic acids, amino acids, proteins, because they use the nitrogen from the nitrate ions. Now, also, bacteria can have a negative impact. So we know bacteria can spoil food. They can cause it to go off or decompose. So things like milk, when they're made and sold in the UK, they have to undergo a pasteurization process, and that involves heating to a very high temperature the milk and allowing it to cool very rapidly just before it's packaged. And that prevents, or that kills, a lot of the microorganisms and bacteria within the milk to below levels that might cause problems when you consume it. Now, we know other bacterial species can be pathogenic. So pathogenic bacteria, quite often when they cause infection in a host, they release toxins. Now, these toxins might damage cells and tissues directly, or they can have other effects. So nice examples here are bacterial meningitis, that infects the linings of the spinal cord in the brain, and it can lead to obviously meningitis. Others are cholera and TB. Now, cholera is quite a nice example because the bacteria that cause that in the small intestine, the toxins released by the cholera bacterium, they bind to a chloride ion channel in the membrane of the epithelial cells that line the small intestine. Now that causes the chloride ion channels to stay open, and so chloride ions end up moving into the lumen of the digestive system. And that's obviously going to drop the water potential, lower it, make it more negative. So water moves out of these cells by osmosis, and it causes a watery diarrhea, which is characteristic of cholera. And that can make you very, very dehydrated quite quickly, and it can quite often be fatal. So we can now start to look at the ultrastructure of a bacterial cell. Now, most bacterial cells are heterotrophic, and that means they actually absorb complex ready-made organic molecules from the extracellular fluid outside them. So that might be molecules like glucose or even lactose, like we've just seen, that disaccharide. Some bacteria are actually photosynthetic, and that means that they don't necessarily absorb glucose from their environment, but they can actually make it through photosynthesis. Now these are called autotrophic bacteria, and the examples are the cyanobacteria. So these cyanobacteria actually inside the cytoplasm have thylakoid discs inside the membranes. You will find pigments that can absorb light energy and convert that eventually into the form of glucose. Now, if we look at the capsule, not every bacterium has this outer kind of slime layer that we call a capsule. And those that do have a capsule, it's really there for protection, because obviously these cells are microscopic. They're very small, normally about 1 to 5 micrometers in length. Now, the cell wall is made of peptidoglycan, and there are two types of cell wall. One bacterial cell wall for gram-positive bacteria will have a very thick peptidoglycan layer. Now, gram-negative bacteria, on the outside of a thinner peptidoglycan layer, will be a layer of what we call a lipopolysaccharide. So there's two main types of cell wall structure that we'll see very shortly. Now, the plasma membrane is a phospholipid bilayer, and within that membrane there are intrinsic proteins. So you might get glucose carrier proteins, you might get channel proteins for certain ions to enter the cell from the outside fluid. So it has similarities with the fluid mosaic model that we've seen in previous topics. It's a phospholipid bilayer with intrinsic and extrinsic proteins within it. There is no cholesterol in the cell membrane of a bacterium because it has structural integrity from the inelastic peptidoglycan cell wall. Right, the cytoplasm then is the main fluid area within the cell, inside which we get many different enzymes that catalyze many different biochemical reactions. So for example, glucose will be broken down through glycolysis in the cytoplasm of the bacterial cell. So all the enzymes for those reactions are located there. Now, the genes that express these different enzymes are located in the main nucleoid, and that is a big circular piece of DNA. And we know DNA is double-stranded. It's made of 2 polynucleotide strands wrapped into a double helix. So all the genes for all the enzymes inside the bacterial cell will be here, especially for things like glycolysis and DNA replication. So that's the main circular piece of DNA free in the cytoplasm. The other circular DNA, it's smaller double-stranded circular DNA called plasmids. Now these quite often will have what we call antibiotic resistance genes on them. Now, it's these plasmids that can be isolated from bacteria and modified through genetic engineering to insert a human gene of interest. So, for example, the human insulin gene could be inserted into a plasmid. We call that the recombinant plasmid, and there are methods to then get the recombinant plasmid into bacteria. And these bacteria, when they divide by binary fission, when they replicate, they also replicate some of these plasmids that then get passed on to the next generation of bacterial cells. Now, on the outside of the bacterium, we can see these pili. So these are actually foldings of the cell membrane. Now, not all bacteria have pili, but those that do, it allows their cell-to-cell interaction. So it allows the bacterial cells to interact, to join together, and sometimes the pili of a neighboring bacterial cell, if we look at the bottom left, will actually make a connection with the pili of this existing bacterial cell. And through this tube, quite often these plasmids can actually move into the adjacent bacterial cell. So this is called conjugation, where plasmids can move from bacterial cell to bacterial cell through the pili that are formed along singular tube. And that's referred to as a type of sexual reproduction in these prokaryotic cells. Now, some bacteria will have a flagellum, or some actually have multiple flagella. And the flagellum has a rotor, so it's a complex of many different proteins within the cell membrane. So this flagellum would actually join to the cell membrane here, and this forms a rotor, and it allows the flagellum to rotate, and that propels the bacterium in a forwards direction. Now, only bacteria that can move, that need to swim in their extracellular fluid, will have flagella. Now, one example of an autotrophic bacterium, like we just mentioned, is the Cyanobacteria. And if you actually look inside the bacterium, what you can see is, although they don't have membrane-bound organelles because they are prokaryotic, they do this time have thylakoid membranes. So these thylakoid membranes are not organelles, so we do get them inside chloroplasts, inside eukaryotic cells like plant cells. But here we've got the thylakoid membranes within the cytoplasm of this prokaryotic cell. So you can see here, this is the thylakoid membranes here inside this cyanobacterium. Now, the photosynthetic pigments are actually located within these thylakoid membranes inside this prokaryotic cell. And they absorb light energy. So you get different types of pigments that might absorb red light or blue light, for example. And they're going to use this light energy eventually to make ATP and also to make glucose. So inside this bacterial cell, in the cytoplasm, you can see the DNA. We can also see many 70S ribosomes in the cytoplasm, the site of protein synthesis. And it has a cell membrane and obviously a cell wall on the outside here. So we can quickly summarize some of the main features of all prokaryotic cells. Now, we know DNA is free in the cytoplasm, and that is either the nucleoid or the plasmids. Now, we know they also have a peptidoglycan cell wall, and it can either be gram-positive or gram-negative, that we're going to talk about in a minute. They have 70S ribosomes for protein synthesis, in particular translation of the messenger RNA that's made against the template strand from either the DNA in the nucleoid or the plasmids. Obviously, the cytoplasm is the fluid-like material inside which we find the ribosomes, and we've got a cell membrane made of a phospholipid bilayer with some intrinsic proteins within that. Now, other bacteria might have their capsule on the outside for protection. They might have one, more, or many flagella, depending if they're motile or not. Some bacteria, like the Cyanobacteria, have what's called photosynthetic lamellae. So these are basically thylakoid membranes with the photosynthetic pigments in them that can absorb light energy that's used in photosynthesis. Most bacteria have plasmids that contain antibiotic resistance genes, but not all bacteria. So some have them, but not all of them. Now, if we actually look at the prokaryotic cells on the left-hand side here versus the eukaryotic cells, so these would be animal cells, plant cells, fungal cells, they're all eukaryotic, and also any organisms that belong to the Protoctista. So animal cells, plant cells, fungal cells, Protoctista cells like the protists that we saw earlier, Paramecium, they're all eukaryotic. So let's just have a quick comparison. Well, we know prokaryotic cells are actually quite small, 1 to 10 micrometers, whereas eukaryotic cells tend to be much, much larger, anywhere up to 100 micrometers. Some plant cells, if you ever do an onion slide and look at the cells and draw the cells, they can be up to 300 micrometers in length, certain plant cells. Now, prokaryotic cells do not have any organelles or membrane-bound organelles within them. The cyanobacteria do have thylakoid discs, but they are not themselves membrane-bound organelles as such. A chloroplast, for example, would be a membrane-bound organelle you'd find inside certain plant cells like the palisade mesophyll. So we know eukaryotic cells do have membrane-bound organelles such as chloroplasts, like plant cells, mitochondria, or fungal cells, and plant cells and animal cells also have mitochondria, and obviously a nucleus. Now, the DNA is free in the cytoplasm of prokaryotes and it's circular. Inside the nucleus of a eukaryotic cell, the DNA is always linear. That means it has a start point and an end point. So all your somatic or your body cells inside the nucleus will have 46 molecules of DNA. And each long molecule of DNA, it has obviously 2 polynucleotide strands in a double helix. That long molecule of DNA is essentially a chromosome. Now, you can't see the chromosomes when the cells are not undergoing mitosis or meiosis. They're arranged as chromatin. So these big long DNA molecules are wrapped around histone proteins to allow them to be packaged inside the nucleus, and that's called chromatin. The chromatin supercoils during prophase, the first phase of mitosis, or prophase 1 of meiosis. So you can start to see the chromosome structures only at the start of nuclear division, mitosis or meiosis. Now, obviously, prokaryotic cells do not have a nuclear envelope. It's a double membrane envelope in all eukaryotic cells that have a nucleus, and it has nuclear pores that allow any type of RNA to exit the nucleus, messenger RNA, ribosomal RNA, or transfer RNA. So all the RNA inside the eukaryotic cell is made in the nucleus and it exits via the nuclear pore. Now, the nuclear envelope might also have ATS ribosomes attached to the outside, and we know the rough endoplasmic reticulum is continuous with the outer nuclear membrane. So prokaryotic cells do have plasmids with their antibiotic resistance genes, and we do not get these in eukaryotic cells. The cell wall of prokaryotic cells is peptidoglycan that we're going to look at in a minute, but eukaryotic cells, animal cells do not have cell walls, but plant cells have cellulose cell walls, and fungal cells like yeast cells have chitin as their cell wall structure. Now, animal cells, they do not have cell walls because as an animal cell is formed, quite often as it kind of changes shape, it becomes specialized and differentiated. So if they did have cell walls, it would not allow the animal cell to change shape. Think about the shape of a motor neuron or a sensory neuron or a muscle cell. So in order to overcome this problem of no cell wall, animal cells actually have cholesterol embedded within the phospholipid bilayer, and that makes the cell membrane less fluid. It kind of makes it firmer, and that allows the animal cell to survive and obviously change shape as it develops and differentiates. Now, chloroplasts obviously are not part of prokaryotic cells. Actually, a bacterium is about the same size as a chloroplast, and it was once thought that chloroplasts that we find inside plant cells many billions of years ago may have actually been independent unicellular organisms on their own, and they might have been engulfed by a slightly larger prokaryotic cell, and because they could actually photosynthesize, they might have had a benefit to that larger cell and therefore became part of it, and that is called endosymbiosis theory. Now, we know some Protoctista have, like the algae, have chloroplasts, and we know plant cells do. Not all plant cells predominantly things like the spongy mesophyll, the palisade mesophyll, and the guard cells, and that gives them their green appearance because chlorophyll, the primary pigment, reflects green light. Now we know prokaryotic cells do not have mitochondria. They might have a mesosome that might be involved in aerobic respiration, but there is still some controversy about whether these actually exist or not, and it might just be an artifact, like we mentioned, of the fixation process to make the slides to view the bacteria. Now we know mitochondria are present in all eukaryotic cells because it's the main site for aerobic respiration where most ATP is made. Now oxygen is needed inside the mitochondria to act as the final electron acceptor, and that allows oxidative phosphorylation where ATP is made. So mesosome possibly present in some prokaryotic cells. There is a bit of uncertainty as if they actually exist or not, but obviously these are not going to be present in eukaryotic cells. Now ribosomes are 70S and they're free in the cytoplasm inside all prokaryotic cells, and they're 80S ribosomes in the cytoplasm of eukaryotic cells, and they're also attached to the outside of the rough ER and obviously the nuclear envelope. Now if we look at the ribosomes inside the mitochondria or the chloroplasts inside the eukaryotic cells, these organelles inside the eukaryotic cells will actually have 70S ribosomes. So there's a number of different ways to classify different bacteria, and the main one is on their shape. And there's three main shapes that you need to know. One is spirillum, and that's shown here on the left hand side. And these unicellular bacteria, these prokaryotic cells, have a corkscrew or spiral shape. Now a nice example is Spirillum minus, and that's the infectious pathogenic bacteria that quite often is transmitted from rat bites that can cause infection in humans. Another nice example of a Spirillum is Borealis that can cause Lyme disease, and that's a tick-borne infection. So quite often in the forests, ticks, insects live on the leaves of plants. If they get transferred onto the skin as you run past the leaves, these insects can feed off the blood, and as they do so they inject the Spirillum bacteria into the bloodstream, and it can cause an infection known as Lyme disease. Now the second type of bacterium is called Bacillus, and that's a rod-shaped bacterium. So we can see here slightly elongated structure, much like a rod shape, and we call that Bacillus. Now E. coli is a nice example of a rod-shaped Bacillus bacterium. The third type is called Coccus. Now that's singular, and COCCI, cocci, that's plural. So these bacteria are spherical in shape. They look rounded when you look at them down a microscope. And the two well-known examples of cocci bacteria are Staphylococcus aureus and Streptococcus pneumoniae. So Staphylococcus aureus quite often can cause conditions like acne, and they tend to grow on the skin. So if we look at the cocci bacterial, these spherical or rounded bacterial cells, we can actually differentiate between Staphylococcus and Streptococcus based on how the cells connect with one another. So if you get a cluster of these round spherical cocci bacteria, like these clusters here, these are actually going to be Staphylococcus. So Staphylococcus aureus is a nice example or species to use, and they're going to cluster together. Whereas Streptococcus, these cells tend to form long chains. So you can see some nice examples here where actually they're not clustered as such randomly, but they tend to interact into a long chain shape. So these are Streptococcus bacteria. So if they're clustered in a sort of random fashion, they're Staphylococcus, and if they're in a chain orientation, they're going to be Streptococcus. And it's really to do with the plane in which they undergo binary fission. So all the cells, the Streptococcus cells that form the long chain, their plane of binary fission is the same, which is why they can form this structure. the plane of cell division or binary fission for these bacteria in Staphylococcus, they tend to be a bit random, which is why they tend to sort of cluster in these groups instead of being in a long chain form. Now, if we actually look at the structure of the cell wall, there's two main structures. Gram-positive bacteria and gram-negative bacteria will have slightly different components. Now, the gram-positive bacteria have a very thick layer of peptidoglycan, and that sits on the outside of the plasma membrane of the bacterial cell. Now, we're going to look at this in a bit more detail shortly, but basically peptidoglycan is made of straight chains of polysaccharide, such as these shown in blue, okay, and these are called peptidoglycan polysaccharide chains. And rather than being cross-linked by hydrogen bonds that we've seen previously in cellulose and chitin, they're actually cross-linked by short polypeptides, or short peptides really we should say, and these short peptides are between 3 to 5 amino acids in length, and it's these short peptides that cross-link the long straight polysaccharide chains, and collectively this forms peptidoglycan. So these short peptides really act like a glue to allow construction of the cell wall. Now, if we look at gram-negative bacteria, there is peptidoglycan on the outside of the cell membrane, but it's a much thinner layer. So if we compare this blue layer at the bottom here to the thicker layer at the top, we can see the thin layer on the outside has an additional membrane called a lipopolysaccharide membrane. So this additional membrane is a phospholipid bilayer, and it has extrinsic and intrinsic, predominantly proteins within it, but it also has additional components. Now, phospholipids are made of a polar head group and 2 fatty acids, and the polar head group of a phospholipid is made of glycerol and a single phosphate group. Now, these lipopolysaccharides are a little bit different. So as the name suggests, lipo, they actually have fatty acids. So they have 2 or more fatty acids, and that's connected not to a polar head but to a longer polysaccharide chain. So these lipids here, as part of the membrane that you can see with this long polysaccharide chain, they're called lipopolysaccharides. So this outer membrane is very rich in lipopolysaccharides, so we call it the lipopolysaccharide membrane. Now, these gram-negative bacteria on the bottom, they are susceptible to antibiotic treatment, but the antibiotic has to be able to get through the lipopolysaccharide membrane, and then through the thin layer of peptidoglycan, and then through the cell membrane here at the bottom into the bacterial cell. And it's similar for gram-positive bacteria at the top. Antibiotics normally have to get through the thick peptidoglycan layer and then through the cell membrane into the cell. However, the gram-positive bacteria are also susceptible to antibiotics that can somehow break down or prevent formation of this thick peptidoglycan layer. So penicillin, for example, prevents the cross-linking between the short 3 to 5 amino acid peptides and the long straight polysaccharide chains in peptidoglycan. So these bacteria treated with penicillin, they can't generate new peptidoglycan cell wall. So the cell wall becomes very brittle and very thin, and these bacteria then become susceptible to osmotic pressure. So if there's no correct cell wall structure, because it's been prevented from forming, when the bacteria take in water by osmosis, the cytoplasm swells and the bacteria burst because there's no proper structural cell wall to prevent expansion of the cell. Now, lysozyme is another enzyme this time, not antibiotic as such. It's an enzyme that we have in our tears, our saliva, our mucus, so it's made naturally from our own cells, and it's part of the innate immune system. And lysozyme actually prevents formation of these long straight polysaccharide chains. It hydrolyzes the glycosidic bonds between these modified beta-glucose subunits, and it actually breaks down existing polysaccharide chains. So lysozyme, this enzyme we actually make as part of our saliva, our tears, and our mucus, that hydrolyzes the glycosidic bonds in the straight peptidoglycan chains, polysaccharide chains, so it also breaks down the gram-positive cell wall. So we can look at the structure of gram-positive bacterial cell walls versus gram-negative bacterial cell walls through a cross-section. And at the top you can see obviously the very thick peptidoglycan layer of the bacterial gram-positive cell wall. Now these long straight polysaccharide chains that we know are cross-linked by shorter 3 to 5 amino acid peptides, so these act as the glue to hold these straight polysaccharide chains together. The enzyme that catalyzes this cross-linking is called transpeptidase. So penicillin, transpeptidase, penicillin is an inhibitor of this enzyme, and it's a competitive inhibitor. So it actually binds the active site of transpeptidase, it blocks its activity, and so it can't catalyze the cross-linking of the short peptides with the straight polysaccharide chains. So as we just said, these bacteria can't make the correct cell wall. Now the reason that they need to continuously make peptidoglycan cell walls is because we know bacteria constantly replicate by binary fission. So when you have a bacterial cell, imagine it was a bacillus, before it divides by binary fission it has to essentially elongate, because it's going to divide to make two cells. Now obviously it does replicate its DNA, and it replicates ribosomes, etc., and then it splits down the middle, and what you'll end up with is two identical bacterial cells. But obviously in order to elongate, the bacterial cell must make new cell wall. So penicillin prevents new cell wall forming, and so the cells become very, very brittle and they're more susceptible to osmotic pressures. Now gram-negative on the bottom, you can see a very thin layer this time of peptidoglycan, and on the outside is the lipopolysaccharide membrane. Now some of these obviously will be phospholipids, the bulk of the lipids in the membrane, phospholipids. It's a phospholipid bilayer. So these obviously here are the fatty acid tails of the phospholipid bilayer. But some of the lipids are actually lipopolysaccharides. So instead of having the polar head group, they'll have two fatty acids or more connected to a polysaccharide chain, maybe like this. And these are actually what we mean by lipopolysaccharides. Now obviously penicillin's not really having much of an impact on gram-negative bacteria, because penicillin cannot readily get through the lipopolysaccharide membrane, and we know penicillin would actually impact upon formation of this thin layer of peptidoglycan, but if it can't get to that periplasmic space, then gram-negative bacteria are not going to be affected by penicillin. So penicillin cannot penetrate that layer. So if we look more closely at the structure of peptidoglycan, what you'll see is that the straight polysaccharide chains actually have two slightly different modified versions of beta-glucose. They have NAG, which is called N-acetylglucosamine, and NAM, which is called N-acetylmuramic acid. So NAG is one type of modified beta-glucose, and NAM is the second type of modified beta-glucose. Now in the straight polysaccharide chains that you can see here, there's alternating NAG and NAM beta-glucose units. So the blue ones are NAM, and then the pink unit next to it here, that is NAG. So these straight polysaccharide chains have alternating modified beta-glucose units. Now if we looked at chitin, the modified beta-glucose that you find in chitin is actually NAG, so this N-acetylglucosamine. So in the straight polysaccharide chains in chitin, you get thousands of repeating NAG subunits, whereas in peptidoglycan, you get alternating NAG and NAM. Now you can see here, actually, you get two short peptides from each chain, and these short peptides are linked actually by a peptide bond. So these short peptides help to glue together the long straight polysaccharide chains, and you can see there's many of these short peptide cross-links that help to glue the structure together, and we know they're susceptible to the activity of penicillin, which inhibits the enzyme that would allow these cross-links to form. So normally cross-links occur by transpeptidase, and penicillin will inhibit that.